Hello again, everybody. I'd like to pick up from where we left off last time. We talked about how plants make starch and sucrose, and how they obtain energy from the breakdown of sugars through glycolysis, the TCA cycle, and oxidative phosphorylation. Today we will focus on oxidative pentose phosphate pathway, and start a new topic, lipid metabolism. First off, oxidative pentose phosphate pathway is this alternative pathway to oxidize sugars in plant cells. It begins with glucose 6-phosphate that is generated from glucose. The conversion from glucose 6-phosphate to ribulose 5-phosphate produces 2-NADPH. Note that here we have NADPH, which has an additional phosphate group compared to NADH that we talked about in glycolysis and the TCA cycle. Both NADPH and NADH are electron carriers. NADPH can donate electrons to help with building other molecules such as lipids. Another important thing is that we lose a CO2 molecule here from the 6-carbon to 5-carbon molecule. Every time you see a CO2 releasing reaction, you should be aware that it could waste the carbon fixed by photosynthesis, especially when the CO2 releasing reaction occurs at a high rate. We can see that the first two steps of this pathway involve oxidative reactions. That's where the name comes from. And these two reactions are irreversible. Next, we have many carbon shuffling reactions that look like the reverse of Calvin-Benson cycle, but they are not the same. Ribulose 5-phosphate is being converted into two different 5-carbon molecules. One of them is ribose 5-phosphate. Ribose 5-phosphate is super useful. It is the precursor to the 5-carbon sugar that makes up DNA and RNA. Then, these two 5-carbon molecules are interconverted to a 3-carbon triose phosphate and a 7-carbon sugar phosphate. Then these two interconvert again to make a 6-carbon sugar phosphate and a 4-carbon sugar phosphate. The 4-carbon sugar phosphate can react with the 5-carbon xylulose 5-phosphate back here to make glyceraldehyde 3-phosphate and fructose 6-phosphate. We know that triophosphates here can go to glycolysis, and conversely, glycolysis can ship triophosphates over here if needed. What kind of benefit are these reversible reactions for the cell? It allows for more flexibility. For example, when cells are actively growing, they need to make tons of DNA and RNA. Then ribose 5-phosphate would be badly needed. These reversible reactions becomes really handy because different sugar phosphates can enter the pathway from different directions to make more ribose 5-phosphate. This is all I want to cover about the oxidative pentose phosphate pathway. The biggest takeaways are, first, it generates the reducing power in the form of NADPH, which can be used for other biosynthetic reactions. And two, it makes ribose 5-phosphate for building DNA and RNA molecules. It's also good to know that this pathway occurs both in the cytosol and the plastids. Now we are moving into lipid metabolism. What are lipids? Lipids are a class of molecules that are highly insoluble in water and soluble in organic solvents. Some lipids are found in the outer layer of plants, essentially the wax-like material on plant leaves, stems, and fruits, which makes them waterproof. This lipid covering is kind of like an extra barrier. It not only helps plants keep social distance from attackers like herbivores and pathogens, but also helps plants retain water under drought or heat stress. Of course, you know lipids are in all cellular membranes, very important for cellular functions. You may also know plant fats and oils are also lipids. Fats are solid at room temperature, but oils are liquid at room temperature. I'm sure you put a lot of these lipids in your mouth or on your skins every day. But for plants, they just want to store energy in their seeds and use them later when the seeds germinate. Coming back to plant fats and oils, they are all made up of the same type of lipid molecule called triacylglycerol, or TAG in short. The head group is a 3-carbon glycerol, and the 3 fatty acid tails are attached to each oxygen atoms of the glycerol backbone. You know, there are different flavors of fatty acid tails in a tag. It can be long or short, straight or curved. This is a fatty acid molecule. The carboxyl group makes it acidic, and the rest of the molecule are just the carbons and hydrogens, also known as a hydrocarbon chain. Each fatty acid differs in their chain length, degrees of saturation, and the position of the double bond. Fatty acids can be as short as 2 to 6 carbons and as long as more than 20 carbons. Usually they contain an even number of carbons. This will make sense when we talk about fatty acid synthesis later. Here we have saturated fatty acids with all single bonds. The molecule is linear, so can be packed tightly with each other, making its melting point high, so they tend to be solid at room temperature. Examples are coconut oil and cocoa butter. Then we have mono unsaturated fatty acids with 1 double bond, like oleic acid. We also have polyunsaturated fatty acid with more than 1 double bond. Linoleic acid has 18 carbons with 2 double bonds. Do you remember the omega numbering system? The first carbon is on the end of the hydrocarbon chain. Then we count inwards 1, 2, 3, 4, 5, 6 till the double bond. So it's an omega-6 fatty acid. Another one is linoleic acid with 18 carbons and 3 double bonds. And this is an omega-3 fatty acids. We need to eat more omega-3 and omega-6 fatty acids because we cannot make them in our body. The double bonds in unsaturated fatty acids create kinks in the carbon chain, making the intermolecular interactions weak, so they tend to be liquid at room temperature. The composition of fatty acids in plant lipids varies with the species. Coconut oils and palm oils are high in saturated fatty acid. Olive, canola, sunflower, and peanut oils are high in monounsaturated fatty acids. Soybean and corn oils are high in polyunsaturated fatty acids. Alright, I hope you have refreshed your memory on lipids and fatty acids. Now let's look at how plants make fatty acids. Fatty acid biosynthesis occurs in the plastids. Because fatty acids are components of membrane lipids in every cell, each cell should be able to make fatty acids by itself. For photosynthetic cells, it is in chloroplasts, but for non-photosynthetic cells, it is in other types of plastids. To kick off fatty acid synthesis, pyruvate is converted to acetyl-CoA, a high-energy 2-carbon molecule. During this process, 1 carbon is lost as CO2, and the reducing power NADH is generated. Acetyl-CoA is the building block molecule for making fatty acids. An enzyme complex known as fatty acid synthase is going to take 1 acetyl-CoA and stick another on it through a series of reactions. Each round of reactions requires 1 ATP and 2 NADPH. We can see the elongation of the fatty acid with 2 carbons. This process is repeated until the fatty acid reaches the length of 18 carbons. That's the maximum length of fatty acids you can make in plastids. These fatty acids can be used to make membrane lipids in plastids and ER. But to make longer fatty acids, the 18-carbon fatty acids are going to move to the cytosol and be extended by an ER membrane-embedded protein complex called fatty acid elongase. Again, you need 1 ATP and 2 NADPH for each cycle. Many of the very long-chain fatty acids with more than 20 carbons are used to make surface lipids. These different lengths of fatty acids can be incorporated into the tags in plant fats and oils, as I mentioned earlier. Lastly, I want to talk about the breakdown of fatty acids, which is extremely important for the germination of oil-containing seeds. At the beginning of germination, storage proteins are degraded into amino acids, which are used to make enzyme. One enzyme called lipase is made to hydrolyze the tag stored in the oil body back to glycerol and fatty acids. The freed fatty acids are going to move into an organelle called glyoxylate, which is a specialized peroxisome in germinating seeds. In the glyoxylate, fatty acids are sequentially broken down into acetyl-CoA molecules. This process is known as the beta oxidation, which involves many enzymes to perform the task. Similar to fatty acid synthesis, each time we are cutting off 2 carbons from the fatty acid, giving us 1 acetyl-CoA molecule. Here we have 14 carbons in this fatty acid, so we're going to cut it 6 times to get 7 acetyl-CoA molecules. Then these acetyl-CoA molecules can enter the glyoxylate cycle. A pathway looks a bit like the TCA cycle in the mitochondria. Here, 2 molecules of acetyl-CoA going through this cycle can give us 1 molecule of succinate. Then succinate can move to the mitochondrion and convert it to malate by the TCA cycle. Then the intermediate malate are going to move into the cytosol and be converted into phosphoenolpyruvate, which is used to make hexose and ultimately sucrose. This process is also known as gluconeogenesis. Okay, we just walked through how germinating seeds convert their seed oil to sucrose. Once they have the sucrose, it can be moved around to support the growth of the young seedlings before they can do photosynthesis. As we come to the end of the lesson, I do want to note that all the pathways in the plant cells are interconnected by molecules. Now, the significance for us is that by changing the components in one pathway, you are very likely to interrupt other pathways. Therefore, it's very important to quantify and predict metabolic fluxes at the network level